Pleasure to be here. It's a bit of a journey to get here today. What I'm going to do in the next 15 minutes is just talk to you a little bit about genome editing and where Syngenta sees the opportunities for genome editing. So we've been manipulating crop genes for millennia. There's nothing new in that in terms of providing the benefits of crossing different plants for increasing yield or bringing in drought stress or environmental tolerance, whatever it might be. We've been doing that for thousands of years. We've been growing these plants in an artificial environment, fertilizers, weeding, etc. So manipulation of crop genes and growing plants in an artificial environment is absolutely nothing new. We've been doing it for thousands of years. And actually, the ability to manipulate crops and manipulate the genes of crops has really been behind this, the increase in yield that we've been delivering to the society as a large since the 1930s, 1940s. So if you look at, this is a global average, this is all crops, looking at a human population global average. Back in the 60s, each hectare of land had to feed approximately 2.3 people. Fast forward that to where we are today, each hectare of land has to feed about 5.5 people. So that's a big increase. We've been able to meet that increase in yield. But looking forward to approximately 2050, there's going to be an extra 2 billion people on this planet. We're talking about 9 billion people on this planet, and that's a huge amount of extra mouths that we are going to have to feed. And actually, it's even a bit more serious than that because of the rise of the wealth and increase in middle classes, such as in countries such as India and China and Brazil, we're going to see a diet moving from a sort of largely a plant-based diet to a more of a sort of a Western-based diet, a diet that we're more used to, the diet that's rich in meat and rich in dairy as well. And those diets are a lot less efficient at getting calories into the human food chain. So actually, by about 2050, we're going to have to increase global yield levels by approximately 50% to meet that calorific demand. So we should really be using every technology that we have at our hands to deliver that incredible challenge, because there is no new land to bring online for agriculture. It's all going to be about getting more out of the amount of land that we have. So I just want to talk a little bit about traditional plant breeding. This is really driven by phenotypic selection. So as an example here, if you have your commercial cultivar and you have a desired gene that you want to bring in from a donor plant, the traditional way has just been crossing these, fertilization, and producing the offspring. But of course, this is going to bring in a lot of undesirable genes that you would not want because it can affect yield, etc. So then to bring back this to the commercial cultivar, I have to do lots of back crossing, and this is, you can do many hundreds and thousands of plants to bring yourself back to where you really want to be in this desired place where you have that desired gene within the commercial cultivar background. That's a very laborious, long-winded way of bringing in that desired gene within your elite line. Newer technologies, in this instance, micro-assisted breeding, can facilitate that process. So micro-assisted breeding really is about identifying regions of DNA and using these as markers in and around your gene of interest, and also in and around genes that you don't want to bring in as well. So then when you cross it with your commercial line, you can scan the DNA at the seedling stage, make a lot more educated guesses about the back crossing you need to do to bring it in to achieve this the desired effect in terms of the gene within that commercial genetic background that you're after. So micro-assisted breeding really allows you a lot smarter ways to select the seeds and do that back crossing to get out the undesired genes whilst keeping in your desired gene of interest. So just to summarize then, for conventional back crossing, if you have your F1 generation, your offspring, you have to do conventional back crossing. You target your gene of interest in the yellow, conventional back crossing with the commercial cultivar to get it right down to really only having in that gene you want to bring in, whilst remaining and conserving your commercial genetic background that you're interested in. Micro-assisted breeding allows you to do that smarter and a lot faster, much smarter choice in terms of back-crossing. But wouldn't it be very nice if we could you'd look at that target gene and just bring it in directly into the commercial cultivar that you want to introduce that gene in? And this is really what genome editing the powerful technique is going to allow us to do. So genome editing is really a technology to deliver that specificity. So it enables this very targeted manipulation of the plant's own DNA. And as Alison said, you can think of it as molecular scissors. You can look at your DNA, identify your gene, and just snip in and snip out that gene of interest. So it gives you this absolute precision. And obviously, it's a huge amount faster than traditional breeding techniques to manipulate that plant gene and bring you that phenotype, that function that you want to bring into your into your commercial line. And actually, the endpoint, the product, can be absolutely identical to that you can generate through conventional breeding. The process is different, but the actual outcome, that commercial line with that gene in, can be absolutely identical to the way you could do it through conventional breeding. So we shouldn't really get hung up on the process. It's the product that we should be thinking about, and the product can be absolutely identical to that achieved through conventional breeding. But obviously, it's a lot faster. You have to do a lot less back-crossing, and you really don't have to worry anywhere near as much about concerns about linkage drag or bringing in undesired genes into that cross. So really, that genome editing is a very powerful set of tools to unlock genetic potential. There are a number of ways to manipulate genes, and genome editing is nothing new. It's been around for 10 or 15 years. The techniques that are protein-based, meganucleases, zinc finger nucleases, and talons, they've been around for a long time, but they've been really difficult to use. You've got to be able to design a protein to interact with a very targeted region of DNA, and that's incredibly difficult. So it's really limited and hampered the take-up of genome editing. CRISPR-Cas is a phrase that you've probably heard. It's really been hitting the headlines in the last couple of years. And this is really revolutionizing genome editing. It's really leveling the playing field, because this technique is so easy. Any molecular biology lab anywhere in the world can use CRISPR-Cas. It's such a simple system to use. So it's only really two components. You have your guide RNA, and you design your guide RNA to be complementary to the region of DNA that you want it to bind to. And you have your Cas9 endonuclease that binds to the guide RNA, and when the guide RNA finds that region of DNA that it's complementary to, it activates the Cas9, and the Cas9 does the job of the scissors and cuts it out. So actually, conceptually, it's a fairly simple process. And if you look at look at this graph here. This is the number of scientific publications with genome editing in. So as I said, genome editing has been around since the turn of the century. But really, it's only in the last couple of years that it's exploded, and that's because of CRISPR-Cas. That's because this technology, CRISPR-Cas, is so easy to use, and it's so cheap as well. And indeed, Nature, one of the leading science journals, has called CRISPR-Cas a disruptive technology. And it's they're calling it the biggest game changer to hit biology since PCR. And PCR, polymerase chain reaction, is the workhorse of all molecular biology labs. And Nature is saying it's going to revolutionize molecular biology just as much as PCR. And indeed, the other authoritative magazine, Science, awarded CRISPR-Cas the Breakthrough Technology of the Year in 2015 because it is so groundbreaking. I mean, we're talking about agriculture today, but also in terms of human medicine. What genome editing can bring to human medicine is just phenomenal as well. So the technology is only just starting, but it's going to be an incredible, make incredible difference to agriculture and human medicine. And really, the benefits of CRISPR-Cas is it's non-protein based, so it's really easy to use, and you can do it very cheaply, so you can just do it at scale. As I said, any molecular biology lab will be able to use CRISPR-Cas. So you can think of genome editing as a set of tools to make a precise change in a genome. We can change a single nucleotide, ultra-single amino acid. We can delete a stretch of DNA or a gene. And we can add or replace a piece of DNA or a gene as well. Right, so this is where I'm going to ask for a bit of audience participation. When Kat first approached me, she said, you've got to try and bring the audience into this. So some of those with an eagle eye might have noticed the dartboard. Yeah. So I'm after someone who likes darts. Who's good at darts in the audience? Come on. Must be somebody. Dave, come on. No one else is volunteering. Come on. Right. So the dartboard is the genome, and Dave's trying to edit triple 20. And he's a conventional breeder. Okay, this is the conventional breeder. So just to make things a bit more realistic. Kat, do you want to spin him around a little bit as well, make him a little bit dizzy? No cheating. So this is the accuracy of conventional breeding. I mean, it's really just laws of nature. Yeah. So that's really how accurate conventional breeding is in terms of being able to manipulate the gene and bring in your desired gene of interest. So now we can step up, and this is what genome editing is. You can step as close as you want. It's that precision. Genome editing he's not very good, is he? But if you're a good scientist, a good scientist will be able to manipulate the gene, that piece of DNA that he wants to with absolute precision, and that's what genome editing is about. So that just brings the point I was making with the dartboard. Conventional breeding really is rather random in nature, whereas genome editing allows you that precise manipulation of the DNA of that gene of interest. So with genome editing, we can knock out one single nucleotide of a gene and alter the function of a protein. We can also delete a larger region of a gene and alter the function of that gene. And we can insert new stretches of DNA, new genes, and we can bring in new functions to crops. And many genes are temporally regulated, whether it's through the course of development, the life stage of the plant, or in response to environmental stimuli. So we'll be able to manipulate gene expression. So we could conceptually upregulate gene expression and bring in the function earlier and faster if we wanted to. And conversely, we can downregulate gene expression and alter the function of the genes and reduce the impact that they would have. So we are starting to see this technology hitting the market now in crops. Not surprisingly, North America is the early adopter of this. So we're seeing soybeans with high oleic acid content. So that's soybeans specifically designed when the oil is cooked to have less of this trans fatty acid, which is a bad fatty acid. So they're designed to have less of that on cooking. Down in the bottom left, we have from a Canadian company, rapeseed canola with built-in herbicide tolerance to the sulfonylurea herbicides, and that's been approved. We have the innate potato. Here they've altered the function of a gene that encodes phenol oxidase. And phenol oxidase causes browning and bruising of potatoes. So this gene, they've altered the function, and these potatoes stay white for a lot longer. And similarly, in the common white mushroom, the phenol oxidase genes, the function's been reduced, and these mushrooms stay white for a lot longer. They don't go brown. So we're starting to see the technology of genome editing and the crops hitting the market now in North America. So I know specifically this audience, you're interested in cereals. So where are we starting to see this cereals? Well, there's no crops hitting the market in terms of cereals. We are starting to see the proof of concept of what genome editing can do. So in wheat, scientists have manipulated and knocked out some key genes, and it's provided resistance to powdery mildew. And we've also seen the concept of proof of principle that genome editing can be done in barley as well, so very precise editing of barley genes using CRISPR-Cas. So finally, just like to end up with just saying how are we using genome editing at Syngenta. So I predominantly work in crop protection research and development. So we are using genome editing to understand the mode of action of new chemistry that we're bringing through the pipeline. So whether it's for insect control, weed control, or disease control, if we've got a new chemistry, we can use genome editing to test a hypothesis about how that compound may be working in an insect pest, a weed, or a fungal pathogen. And resistance, that's a big concern for us and for yourselves. When we start to see resistance in the field, we can test hypotheses about what the molecular mechanisms might be behind that resistance by using genome editing. So this is how we are and will be using it in crop protection research and development. And more widely, looking at the Syngenta portfolio, Syngenta operates and has research in all of the key field crops, rice, corn, soybean, and the cereals. And we also have a broad portfolio of diverse field crops, such as spring onions, sugar beets, specialty crops, such as potatoes, coffee, etc., vegetables, sugar cane, and we've got a large lawn and garden business with a lot of flowers as well. And we see the potential of genome editing to bring in desirable traits, new solutions, whether it's resistance to pathogens, increasing yield, new colors for flowers, whatever it might be. There's the potential for genome editing in all of those plant species. So just finally, just to wrap up, hope you've got across to you today just how powerful genome editing is and how simple a technology it really is, and the power that technology is going to bring. Of course, the thing that's really going to govern this technology is the understanding of crop genetics, the understanding of the biochemical pathways, understanding how when those biochemical pathways are expressed and how they interact with the environment. So that basic understanding is still going to probably limit the application of genome editing technology. So there's a huge amount of basic plant science we still need to be able to do to really open up and fully exploit the doors here. But clearly, as I hope you can see, it's going to make a huge difference to our crop protection research development, and you will start to see new crop and plant trait solutions brought to the market through genome editing. So that's where I'd just like to finish my talk, okay?